All right. Hello, everyone, and thank you all for joining us during the Lytham Partners Spring 2023 Investor Conference. My name is Robert Bloom, Managing Partner of Lytham Partners. During this fireside chat, we welcome Shuttle Pharmaceuticals, ticker symbol of SHPH on the NASDAQ, and their CEO, Dr. Anatoly Drachilo. We'll dive into questions in just a moment, but I want to remind everyone that management is available for one-on-one meetings later today. If you have not already signed up and would like to do so, you can send me an email at bloom@lythampartners.com or visit our website, lythampartners.com/virtual. Click on the one-on-one meeting request button, and we'll look to get you taken care of. So with that, let's begin. Dr. Drachilo, thank you for participating today.
Yes, good afternoon. How are you doing?
Doing good. Before we begin, could you just provide a little bit of background on yourself?
So I'm a physician, radiation oncologist. I worked for many years taking care of patients and running the Department of Radiation Medicine at Georgetown University School of Medicine. I've also served as Cancer Center Director, interim for the Lombardi Comprehensive Cancer Center. So most of my work has been with cancer patients, the treatment of cancer patients, and with research to improve the outcomes for patients.
All right.
The company was formed in 2012, where we had several interesting technologies that we wanted to advance to clinical applications, and the company itself, as a private company, developed new technologies, including new inventions of HDAC inhibitors. We then later on filed for some funding from the government, from the SBIR program at NIH, and we were able to complete a phase 1 clinical trial with our candidate lead clinical drug, ropiduxuridine, which is a radiation sensitizer for improving radiation treatment of cancer, and also for the HDAC inhibitors. We developed a number of platform technology and a number of patents for various applications of these drugs.
So as we've sort of touched about in the past, Shuttle's mission is to improve the lives of cancer patients by developing these therapies that you mentioned that are designed to maximize the effectiveness of radiation therapy while limiting the late effects of radiation in cancer treatment. For sort of the lay person out there, help us understand what exactly this means.
Yeah, so the technology for doing radiation therapy has really come forward in the last 20 years and is outstanding in that we can deliver any dose we want to any part of the body. The limitation that comes in now is the fact that if you give too high a dose to areas where the normal tissues are sensitive, then you damage the patient and you cause harm. So the idea that we could deliver safe doses of radiation by using sensitizers to help enhance the effect on the cancer cells as opposed to the tumor cells, that's a concept that helps us to improve the outcomes of this technology. And radiation therapy is used in treating many, many cancers. So radiation therapy can be a primary modality where we just give radiation and that's the primary treatment that's necessary, or it can be in conjunction with either drugs or with surgery. Here, what we have been focused on is to make the treatment better for patients, to have fewer toxic events so that patients have fewer side effects, and to cure more patients, obviously.
So you've developed not just one, but a complete platform of sensitizers here. Let's first discuss your lead candidate, ropiduxuridine, which you touched on in your introduction there a bit. Can you discuss how it works and your clinical indications?
Right. So there's a number of ways that you can approach the radiation sensitization or improvement of the radiation effects. One way is to upfront to make the cancer cells more sensitive to radiation. And that's what ropiduxuridine does. It's a drug that is taken by mouth, and then it gets incorporated into the bloodstream and gets into the DNA of actively growing cancer cells. By incorporating into the DNA, the DNA becomes more fragile to the effects of radiation, so we're able to get more damage, more double-strand breaks, and we're able to treat the cancer cells to a greater amount than any normal cells. Normal cells usually are not actively growing. They're basically slowly turning over, and they do not take up the drug. Now, on the other end, we also have developed the HDAC inhibitors, which help us to stimulate the body's own immune system to attack the damaged cancer cells that may still be there in the tumor that have not been destroyed. So this is a two-way platform that we developed overall, sensitized to the direct effects of radiation, and then to stimulate the innate immune system to attack those cells that need additional, damaging effects from the immune system. By doing this, we think that we can balance the toxicities versus the positive effects of radiation.
Maybe talk a little bit about the competitive landscape for sensitizers used with radiation therapy here.
Right. So there really is only one drug that was actually approved for the indication of radiation sensitization as a primary indication. And that drug is a biologic. That's an antibody that has to be injected. Then it has been used in head and neck cancer. All the other drugs that are currently used are basically drugs that are used off-label. The drugs that were initially approved for other indications, for the treatment of GI cancers, for the treatment of head and neck cancers, for lung cancers, and basically they have a side effect of radiation sensitization, and those drugs are used at the discretion of the physicians. The drugs that we're proposing have no other effect. So the ropiduxuridine is, if you give it to the patient, you have no effects unless you irradiate. So these are true radiation sensitizers in that category of drugs. And what happens is that by not having those other single agent effects, you would decrease the number of side effects that would be available. And then, of course, you'd have a specific treatment and a more personalized treatment for patients who receive radiation.
Could you expand a little bit on the market opportunity and, sort of circling back to the indications here for ropiduxuridine?
So the as a physician, I see the market as basically patients that need additional improvement in their treatments. So in our situation, the ropiduxuridine, we're targeting developing this for the treatment of brain tumors, which is one of the most devastating types of tumors. The brain tumor that we've picked is the most aggressive, the glioblastoma multiforme. And we picked a tumor that does not respond well to any other drugs or treatments. And this is one of those situations where if it's a rapidly growing tumor, takes up the drug, we can make the radiation more effective in our treatment. Now, overall, the market is pretty large. There's about, anywhere from, 1,800,000 patients, that are diagnosed with cancer in the United States. And out of those, there's about half those patients will have treatment for localized disease, or let's say with the curative intent. And in that group that we look at, the subset that would benefit from a sensitizer is about half of those. So we're talking about somewhere in the range of about 400,000 to 450,000 patients overall for whom radiation sensitizers could be developed. This is a fairly large market. And I think that in today's environment of personalized medicine and what they call precision medical care, there will be other drugs that will be developed for various indications that involve radiation therapy. From the point of view of brain tumors, there's about 10,000 patients diagnosed with brain tumors every year. And as I said, very few of these patients can be managed surgically only. They're usually combined with other treatments. And certainly the addition of a sensitizer would play a big role in that group of patients.
With respect to ropidexuridine here, discuss the upcoming sort of next steps and milestones folks should be looking for.
So the phase 1 clinical trial has been completed at Brown University, and with government funding to get this done. And it looks like the dose has been fairly safe in patients, meaning that there were some toxicities, but there was a fairly reasonable dose that can be given to patients and gets into the blood. And certainly was we were able to complete the clinical aspects of this trial so that we can move to the next level. That trial established what's called the maximum tolerated dose, which is the dose that's then used to advance next. The next dosage level will be 1,200 milligrams per day, or 1.2 grams. And this is something that can be given to patients daily for up to a month. The clinical trial, they tested for 28 days of dosage, and it was tolerable in patients, and certainly was not a problem. So our development would focus on doing the phase 2 clinical trial to see if it can improve the survival of this very aggressive subset of brain tumor patients. If that is something that we are successful in completing and seeing a benefit, then we would then expand to a randomized trial that would allow us to seek commercialization of the drug.
All right, great. As we've discussed a bit, you've developed sort of this whole platform. So beyond ropidexuridine, you have your HDAC inhibitor platform. You've discussed here in some preliminary detail. Could you maybe expand on that program a bit more broadly?
Right. So the HDAC inhibitors is something that I've worked on for about 25 years in terms of doing research. And there are several HDAC inhibitors that have been out there and marketed already. Merck has a drug called vorinostat. But these are what's called pan-HDAC inhibitors. And pan-HDAC inhibitors affect all 12 of the enzymes that are part of the HDAC group of targets. These are enzymes that do various things like change the conformation of the DNA. They affect the growth, cell cycle, and all the very basic, aspects of cellular function. And the problem has been that if you affect too many of those processes, you get a lot of toxicities and unwanted, effects on patients. So the focus of most research has been to identify the specific HDAC subtype that would affect a certain function in the cell that is useful. So we have targeted the class 1, HDAC molecules for sensitization. So there are, four enzymes that we target. And the class 1 allows us to sensitize certain cancers like squamous cancers of head and neck origin. And we've tested that in the laboratory. We also tested, some class 2, and specifically the class 2 enzyme that's of greatest interest is something called the HDAC6, the histone deacetylase number 6. This molecule regulates the innate immune system, and it's involved in changing the effects of certain white cells in terms of how they respond and how they stimulate the immune system to attack different targets. It's part of your inborn resistance to various infections and injuries that occur from the outside. So we feel that by combining it with radiation, we would be able to target cells that may not have been completely destroyed by radiation because of the lower doses we give, but we know that by radiating cancers, we stimulate the antigens that are produced and go to the surface of some of these cancer cells, and those become the target for the immune system to attack. And that's the basic overall strategy. And we'd like to develop that for specific tumor types. We're particularly interested in tumors such as breast cancer. We're interested in, lung cancer. These are all areas that we will be investigating, to develop phase 1 clinical trials and then to advance to a subsequent phase 2 with our new drugs.
And maybe any sort of, I know you just touched on them a little bit there, but any of the upcoming milestones and next steps within the HDAC platform that folks should be keeping an eye out for?
Right. So the drugs have been discovered and tested in. So some have been tested both in cells and animals. Others have been tested in cells. The HDAC6 inhibitor that we have is, we're pretty high on because it has been tested in animals and shows specific effects on cells called macrophages in terms of changing the dynamics of how they train T cells to attack cancers. So this is the group that we're most focused on. And what we will do is the milestones that we're looking at is to complete the IND enabling studies. And as we complete the IND enabling studies, we'll then be able to get into a phase 1. And I'm looking at that for about a year and a half down the line to be able to get to that point.
Okay, gotcha. You recently went public. Can you discuss the cap structure, balance sheet, and maybe any guidance that you've provided to the street as to how far out your cash will fund operations?
Right. So the one thing I discovered in becoming a CEO of a company coming out of academics is that you spend all your time thinking about how to keep the company extended on their platform so that there's enough money to make sure everything that you want to do can be done. And this is something that we have, our initial raise allowed us to have sufficient funding for the clinical development of ropiduxuridine through phase 2. Then we've also followed this up with a secondary raise where we raised another $4 million for the IND enabling work that will be done with the HDAC6 inhibitor. So our pathway right now is that in terms of the financing that's available is sufficient to go to for the duration of the phase 2 clinical trial to get well into that. So we're talking about a year and a half to 2 years with the current financing, and we will be seeking additional funding along the way as we reach some of our milestones.
Okay. Maybe in the couple of minutes that we have left here, talk a little bit about the intellectual property surrounding the platform as a whole.
Right. So the ropiduxuridine is a drug that has been basically developed with government funding, taxpayer dollars, and it's something that was available to us under a FDA granting of a letter that says that we have orphan designation for this drug for brain tumors. Right. So that has some potential, tax advantages, clinical trial advantages, and also protection for marketing for up to 7 years. The other drugs, the HDAC inhibitors, were all discovered and invented in our laboratories of Shuttle Pharmaceuticals. We have patents on about a half a dozen molecules, and the ones that have been awarded already are listed in our on our website. So we have the patents that are available. The ones that we will target, we'll develop further. Others we will try and develop some partnerships with, where we can maybe get a synergy with other companies.
All right, perfect. Maybe in the couple of minutes that we have left here, anything that we haven't touched on that you think would be helpful to folks here?
No, I think this is an area, radiation sensitization, that has not really been explored much, at least with other companies. And part of it is because these are very difficult, let's say, trials. You have two things you have to do. You have to have a drug and you have to have radiation. And you have to identify areas where you can show a benefit that's acceptable to the FDA. Radiation is effective, so you have to treat well enough with a drug to improve the outcomes. And really, it's an area that I think will be explored much more as we go forward, and we start developing these specific targeted sensitizers. It fits very nicely with the development of the immune system in terms of treating cancers and personalized medicine.
Perfect. Sounds good. Well, thank you so much for your time today. Greatly appreciate it. Again, I'll just sort of remind everyone that if you've not already signed up for a one-on-one meeting and would like to do so, reach out to me, Blum, B-L-U-M, at lythampartners.com, or again, visit the website, click on the one-on-one meeting request button. We'll look to get you taken care of. So we hope you all enjoy the conference. Dr. Chillo, thank you so much.
Thank you for having me here. Thank you.